Hello everybody, in this video I'm going to be talking about the process of osmosis. Now this really fits in with the other topics like diffusion and active transport in the section of the syllabus really known as movement across cells. Now osmosis really refers to just movement of water, and we'll come to a definition in just a moment, but I think to start the video I'm going to start in the same way that I start with my own students in the actual lesson, and I show them this image and ask them, why do they think feet wrinkle in the bath? It's a really interesting question. So exposure for like 5 minutes to water and you get this kind of wrinkling effect on the soles of the feet, on the palms, on the fingers. And usually I get the response of maybe water's coming out of the skin, out of the cells, and that's why it's appear wrinkled. In fact, recent studies suggest that it's more of a behavioural adaptation for gripping. But it's actually not about water moving out of the skin. In fact, the actual fatty layer of the skin, because ultimately cell membranes are made of lipid, prevents water from moving in and out very easily. Now, the reason why I'm starting with this particular question is because this idea of moving water in and out of the body or between different cells is really key when we think about osmosis. Osmosis is really critical in maintaining the correct water levels in all parts of our body, specifically in our cells. If there's too much water or too little water in one of the cells, then that can have quite devastating consequences, as we'll come on to. And we'll come back to this wrinkled feet idea in just a moment. So let's look at a definition. I've said in a previous video what diffusion is. Diffusion is the movement of particles from an area of high concentration to low concentration down a concentration gradient. It's almost like moving things from A to B, from where there's a lot to where there's a little. Now, diffusion purely of water, so we're specifically talking water, nothing else. That's what we mean by the term osmosis. So we only refer to the term osmosis when we're talking about water. So it's the movement or diffusion of water from a region of high water potential. Now notice we're not saying high concentration, we've just swapped the word. So this term water potential we use instead of concentration when we're talking about osmosis. So it still means ultimately where there's a lot of water. Almost, when I like to think of it, when I say high water potential, as in the potential for there to be lots of water there. So from the region of high water potential to a region of lower water potential. So again, ultimately it's the movement of water from where there's more of it to where there's less of it. Almost like a high concentration to a low concentration. Now in brackets, I've put a little extra detail that might help. So when we think higher water potential, we're referring to a dilute solution. Because if there's lots of water there, it's likely that that solution is very dilute. Where there's a lower water potential, so where there's technically less water, that solution would be much more concentrated. And that's particularly important when we come to think about exam questions, even like this question I'm going to talk about in a moment. So osmosis, the diffusion of water from a region of high water potential to a region of low water potential, and what's really crucial is this last part, through a partially permeable membrane. Now, one of the exam questions that I really like asks, why not the word semi-permeable? Semi means half, and it implies half of the things can pass through this membrane. And that is incorrect. We use the term partially. Partially means some. It lets some things through and not others. So let's have a think at the bottom, and I know this looks a little strange, but this little cartoon version would be at the bottom. So we're going to have a think here at these 3 bullet points that I've got. And I've got a picture here of a sort of container. So this is meant to represent almost like 2 cells. I've got a container here, you can see, divided in the centre here by this partition. And this is going to act as our sort of membrane. We've got water and sugar solution in these containers. And we can see that we've got water in blue, sugar in red, and you can straight away see that there's a lot more water on the left side of this image than there is on the right. On the right, there seems to be a lot more sugar. So let's answer these particular questions. So where is the semi-permeable membrane? So the semi-permeable membrane is right here in the middle. So this membrane, if I just draw a more solid line, this is going to let some things through and not others. It says here on the left, which side is more dilute, so it's the higher water potential. Well, clearly, if there's more water on, if we call this side 1, this side 2, if there's more water on side 1 has the higher water potential. Side 2 is the more concentrated, sugar concentrated solution, so that's more likely to be our lower water potential region. And then finally, which direction will osmosis happen in? Well, if we go to the definition, it moves from a higher water potential to lower, so water would move from 1 to 2, as the arrow suggests. There's technically more water on this side and less water on this side, so the water will move across. And that really is it. That's what osmosis is, the diffusion of water from where there's more, ultimately to where there's less. Now, just a really crucial thing to consider here. The sugar is, in this image, dissolved in the water. It's a sugar solution. Notice we've used the words at the top, solution here and solution. The reason why that's important is because certain things in plants and animals don't dissolve in water. If we take a really good example in plants of starch, starch is insoluble in water. It doesn't dissolve. Anything that is insoluble in water will not affect the water potential. It doesn't affect what's called the osmotic potential or osmotic gradients. So that's just something to bear in mind. The reason why plants deliberately store glucose as starch is not only because it's compact, but because it is insoluble, so it won't affect water potentials. So in questions about osmosis in the exam, you just need to make sure that if it's referring to a particular solution, it's talking about usually things that have dissolved in the water. And it's not those that move. So in this example, it's not the sugar that's moving at all. It's the water that is moving. Osmosis is just referring to diffusion of water. What we've got here in this image is something known as plasmolysis, which we're going to talk about in a moment in a bit more detail. But if we have a look, what we've got here are plant cells, and if I just highlight the cell wall, just very crudely, so with an arrow. So here we have a cell wall, and here where this other arrow is pointing to, this outline, this is pointing to the cell membrane, and you can see it's been pulled away from the cell wall. Now that's a particular phenomenon that you get when water is being pulled out of a plant cell. So when water moves out of a plant cell, we can see that that cell starts to shrink and it can pull the cell membrane physically away from that cell wall. So what we're going to do now is look at particular animal and plant cells and talk about whether, because of osmosis, whether those cells would increase in size and swell or shrink. So what we've got here are some pictures at the bottom of animal cells and plant cells. So the plant cell, the example we've got is just a typical, we're going to go for a palisade, but it's a mesophyll cell, and that animal cell at the top, for example, is a red blood cell. Now, I'm going to start with the column in the middle. Now, the column in the middle is looking at the water potential of the solution outside. So what we're doing in this scenario is imagining that we're going to take these cells and put them into a solution. Now, just for ease, imagine if you could pick this cell up and put it into a beaker of water. Let's just say, for the sake of argument, that we can put this beaker into these cells into a beaker of some kind of watery solution. So what we're looking at is, what is the level of water like in these cells compared to the water level outside of the cell in this solution, in the beaker, if you like? I mean, in real life, we think about it in terms of the body or in terms of the plant, but it might be easier to think of it like that. So water potential out of this solution outside, we're going to go for the middle column, and we're going to imagine that it's equal to that of the cell. So we've got below net movement of water in or out. Well, if the water potential is the same, that would ultimately mean that there's no water movement. So we can put here nothing. There is no water moving in or moving out because our water potentials are equal. Equally, if we look at the state of the cell, as in if our options are normal, swell or shrink, because water isn't moving in or isn't moving out, once again we're going to put nothing there. So it would appear completely normal. Now let's take the left column where it says higher than in the cell. So that is saying that the water potential of the solution outside is higher than the actual water potential in the cell. Going from our definition of osmosis, that water moves from an area of high water potential to lower, if it's higher outside and lower in the cell, then hopefully you can see that water would move in. Water moves into the cell because it moves from where there's a higher outside into lower, which is inside the cell. Now, if water moves in, then what we see is that the cell begins to swell. And that's what we can see in these images here. So on this one here, this red blood cell, you can see looks a lot more swollen. You've got this characteristic, if I just draw a little ring here, that sort of dented biconcave shape of the red blood cell. We've lost that on this picture to the left here. It looks like a swollen cell. Water has moved into that cell. If we look at the plant cells at the bottom, we can see something quite similar. The cell looks like it has swollen ever so slightly. Now let's look at the right-hand column, lower than the cell. So that's saying that the cell has a higher water potential than the solution outside. There's more water in the cell than there is outside. And if that's the case, water therefore would move out. And as you can imagine, as you can see in these images, this red blood cell here, and this plant cell at the bottom here, which we're going to talk about in a bit more detail in a moment, that would cause these cells to shrink. So there we've got some examples of what happens to particular cells when they're placed inside of solutions of varying water potential. Now, certain exam questions will ask you this kind of question. They will give you maybe a picture of a red blood cell or another kind of type of cell in some kind of solution, and it will tell you where there's more water, and it will ask you to describe what is happening to the cell. And if there's more water in the cell than outside, the water will come out, and so the cell will shrink. And if there's more water surrounding that cell, water will move into it, and the cell will swell. Now, the words swell and shrink, we really reserve for the animal cell. The plant cell is a bit different. There are a few key terms, a few fundamental things to say about that plant cell. Now, depending on the specification that you're studying will depend on which of these statements would be most relevant, but I think it's important to consider all. Now, plant cells don't burst when water comes into them. That's because water pushes against the cell wall, which is called, or classed as inelastic. So we're just going to add a few notes here. So this cell wall here, we describe as being inelastic. So, as you might suggest, as in not very elastic, not very stretchy. And what that does, when water moves in, so when we fill this cell with more water, it kind of pushes against the cell wall. And being inelastic, what it does, it creates pressure. And we call that pressure Turgor pressure. So we're just going to make a note of that in brackets here. So that I've just described is known as Turgor pressure. Now, when that happens, in the table at the top, I said the word swell. You can use a specific word for the plant cell to mean swell, and that word is turgid. So we can say that the animal and plant cells would swell if water moves into them, but we could also use the word turgid when referring to plant cells, and that means water's moved into them. They're a bit bigger than normal. Now, when a cell is placed in a solution that has exactly the same water potential as itself, so the same concentration of water, there's no net movement, and we've seen that at the top. There's no net movement, nothing happens. When a cell is in a more dilute solution, so when there's more water and less solute, for example, that's when water moves into the cell, and we've said it becomes turgid, and the vacuole, so this area that I've highlighted in blue, the vacuole, we've said that swells. But now we need to consider what happens on the other side of this kind of table. What happens when water leaves? And this links back to the idea of plasmolysis that I was referring to. When a cell is in a more concentrated solution, so when there is more solute, so like sugar or salt, and less water, water will move out of the cell and it becomes what's called flaccid. So again, we can use the word shrink to describe these cells, but particularly for a plant, we can use the word flaccid. Now, at that point in a plant cell, something really distinctive happens. The cells start to contract and become damaged. The cell membrane pulls away from the cell wall, and if I draw this in red, you can see here where I'm drawing these red arrows. This cell membrane has pulled away from this outer cell wall that I'm highlighting in red here. And that is known as plasmolysis. So we'll just put that term in here. So there we have it. A little bit about osmosis, the definition, the movement or diffusion specifically of water from a region of high water potential to a region of low water potential through a partially permeable membrane. We've got some descriptions of animal and plant cells and what happens when inside different or varying water potentials of solution. And one thing to just come back to, something that I said about the skin and the opening question about why the skin wrinkles. Just wanting to think, what would happen if the cell membranes didn't control actually what comes in and comes out of the cell? The job of the cell membrane is to control what comes in and what comes out, but if that didn't happen, then potentially osmosis would happen and water would move in and out. And if that could happen, if you're in a bath full of water and your cells have less, then technically all of your swells would burst. So you can see it's pretty important that your cells have that kind of water impermeable layer. So some key things: water moves in and out of the cells by osmosis through the cell membrane. It is an important solvent, i.e. it lets things dissolve in it. A good thing to remember is that most cells contain about 75% of water, and the reason why it's so important is because water transports substances and allows many chemical reactions to take place. So therefore it is critical that our body controls osmosis and osmotic gradients as much as we can. Okay, hope all that helps.